Hello everybody, welcome. Today we're going to be discussing carbon-based molecules. And first we need to have a little discussion about carbon itself. So carbon is an element which has 4 valence electrons, which means that it can form 4 different bonds for every carbon molecule. And basically what that means is there's this wide variety of potential bonds and potential elements that carbon can bond with. It can make single bonds, double bonds, triple bonds. It can make ring-shaped bonds. It's the most versatile element. You can literally make millions of different structures, which is why carbon is the basis for all living things. You can see just a couple examples here. 4 single bonds here. Here's a triple bond and a single bond. Something you'll notice is every single time carbon makes 4 different bonds. Same here with this like ring formation. You can see this carbon makes 1, 2, 3, 4 bonds, and so on. So all the molecules that we're going to be talking about in these notes are carbon-based macromolecules, which means they are carbon-based giant molecules. And when we talk about macromolecules, it's this giant molecule that's made up of smaller molecules called monomers. And you string a bunch of monomers together, you get a macromolecule. Macromolecules are also known as polymers. So they're kind of 2 words that basically mean the same thing, polymer, macromolecule, both made up of monomers. Here's some example. Starch and cellulose are macromolecules. Its monomer is glucose, so you string a bunch of glucose together. DNA and RNA are macromolecules, and its monomers are nucleotides. Same thing with proteins, and its monomer is nucleic acids. So there are 4 groups of macromolecules we're going to talk about: carbohydrates, lipids, nucleic acids, and proteins. So let's go through each one of those, give you some basic understanding of each one of them. So carbohydrates are always made of carbon, hydrogen, and oxygen. And it's always in a ratio of 1 carbon to 2 hydrogens to 1 oxygen. Example, glucose is C6H12O6. So 6 carbons, 12 hydrogens, and 6 oxygens. Carbohydrates are used as the main source of energy for life. Carbohydrates are the preferred macromolecule that gets broken down and turned into ATP in the body. Some examples: cellulose is a macromolecule that's used as a structural polymer in plants and makes up the majority of its body and skeleton. Starches are how plants store energy. Well, it's a little different in humans or animals where and excess energy is stored in fats. Carbohydrates is probably the most complicated when we talk about monomers, because the monomers can be either simple monosaccharides like glucose, galactose, fructose, or disaccharides like sucrose, maltose, and lactose. Anything that ends with the word ose, like glucose, galactose, fructose, those are all sugars. And how these carbohydrates work is you string together multiple of these either single sugars or double sugars until you get a long chain, like cellulose. Let me just show you. Here's another example of a large carbohydrate. This one is called glycogen. Glycogen is used in animals, so in your body, to store sugars for a little bit longer of a time. So we're talking hours to days here. So that's carbohydrates: carbon, hydrogen, oxygen. The monomers are monosaccharides, which means single sugars, or disaccharides, which means double sugars here. But in either case, whichever one you use, they get strung together to make larger molecules, which are known as the larger carbohydrates. The second macromolecule we're going to talk about are lipids. Lipids are used to store energy in animal long term. They are also important parts of biological membranes. Think of the cell membrane and how it's made of a phospholipid. They are made almost entirely of carbon and hydrogen. There's a little bit of oxygen here and there, but generally it's almost all carbon and hydrogen. They are not water soluble. And a few different categories includes fats, oils, waxes, and steroids. Two examples that I want to differentiate for you are called saturated fats and unsaturated fats. So in a saturated fat, each carbon atom in the fatty acid chain is joined together by a single bond. Here's what that means. Here is the fatty acid chain. So lipids are made of glycerol, which is right here, and a fatty acid chain. Boom, boom, boom. So every single carbon in the fatty acid chain in a saturated fat is single bonded here. Okay. And what that means is that those macromolecules or those atoms can get closer together. And closer together, molecules can form van der Waals forces. Remember, we talked about that way early when we talked about like chemistry. So those van der Waals forces are slight attraction between different molecules. What that basically means is that the molecules in the fatty acid chains are more attracted to each other, so they're more likely to be solid at room temperature. Things like butter, margarine, lard, shortening, they're all examples of saturated fats. Now, unsaturated fats have more than one or more double bond in the fatty acid chains. So what that means is these molecules can't basically get as close to each other as in a saturated fat because there's like little kinks in this chain. And that means that there's not as many van der Waals forces, and these things are liquid at room temperature. Basically, all of the oils you can think of, olive oil, peanut oil, coconut oil, any cooking oil, those are all examples of unsaturated fats. Next example of a macromolecule is called a nucleic acid. The function of nucleic acids is to store and transmit genetic information. And a cool thing is that every life form on Earth uses these same types of nucleic acids, and those are RNA and DNA. Nucleic acids are made of the monomer called nucleotides, and here is a single nucleotide. The nucleotide for nucleic acids, or for this one, let's just say it's DNA, would be a nitrogen base, a 5-carbon sugar, and a phosphate group. And so you string these molecules together to get a larger molecule like DNA. And the difference between DNA and RNA, at least one very basic difference in the monomer, is this 5-carbon sugar. And so in RNA, it's a simple ribose sugar. Ribose ends with an O-S-E. That means it's a sugar. While in DNA, it's a deoxyribose sugar. So really, it's just missing an oxygen from ribose. Here's the sugars, right? Ribose, deoxyribose. The only difference there. And so we have nucleic acids. Function is to transmit genetic information made of carbon, hydrogen, oxygen, nitrogen, and phosphorus. Here is what the monomer looks like, and that's called a nucleotide. All right, moving on to our last one, which is proteins. Proteins have a few different functions. Proteins can control the rate of reactions, chemical reactions in the body. They can regulate certain cellular processes. They form bones and muscles, transport substances in and out of the cell, fight diseases. Proteins do a bunch of different things. So a protein is made of carbon, hydrogen, oxygen, and nitrogen. If the protein is the polymer of the macromolecule, they are made up of amino acids. So amino acids are the monomers. Amino acids all have the same general shape too, where one side has an amino group, which is this NH2, and the other side has a carboxyl group, C double bonded to O and H. Let me show you what that looks like. So here is a single amino acid where it's got an amine group, so this N, 2 hydrogens over here, and a carboxyl group on this side, and then a central carbon attached to an R group. And that R group can be anything. There are 20 different amino acids, and they only differ on what's in their R group. So here's one amino acid called glycine, and its R group is just a simple hydrogen. While this alanine, its R group is CH3. And all that differs between each of these amino acids is what's in that R group, okay? Then when you make proteins, this is the bond that happens where it's called a peptide bond. You can see here. The amine group from one side bonds with the carboxyl group of another side. And when you get proteins, you have these strings of amino acids strung together. And then it goes through a series of folds and twists and stuff to make a protein which has a complex function. But they all generally start as simple as this, where you have one amino acid bonded to a second amino acid and to a third and a fourth and a fifth and so on until you get this long chain of amino acids. Okay, so that was our notes. Make sure you understand the difference between the 4 types of macromolecules, what a macromolecule is, and all of that. Thank you for watching.